Hi. One of the powerful features of MiXCR is the ability to perform quality control. And today I'm going to show you how easy you can create quality control plots and examine them. We're going to start by opening the terminal, and we're going to go to the directory that we've used in one of the previous videos. Here in the results folder, we have an output files generated by MiXCR analyze command for 3 samples. So without further ado, I'm just going to go and create the first quality control plot, which will represent the efficiency of alignment. First, I'm going to provide the path to the MiXCR directory. Now, the name of the function is export QC. Then the name of the metric is align. And finally, I have to provide the input files, which we have in the results folder. Now, you can use both VZJCA files or CLNS. Both of them have all the information needed to perform the analysis. So I'm just going to go and type star.clns, which means take all of the CLNS files in the results folder and align qc.pdf. Now you can also use other output extensions like EPS, SVG, PNG, or JPEG. Here I'm just going to use PDF. And now we just open the file that was generated just now. And here we go. Here is the plot, which represents the rate of alignments. So here we see the percentage of reads that were successfully aligned or not aligned due to various reasons. We see that information for all of 3 samples. And what we can tell from here is that 90% of the reads have been successfully aligned, which is the good quality. Then there's small portion of reads that has not been aligned, mostly due to absence of TCR or immunoglobulin sequences, which is said in the legend that there are no hits. And for further investigation, one can use MiXCR to export those reads and manually inspect them to track their origin. Now, the next metric is called tags. So I'm gonna start by using the same command. I'm just gonna delete all of that and say tags. Again, the input files can be either VZJCA or CLNS. I'm gonna use CLNS. And the output file name is tags.pdf. Now I'm gonna open this one. Now, tags report will vary depending on the preset. It represents the filtering based on different barcodes. This preset was created for Takara Bio BCR kit, which has UMIs, and MiXCR performs an automatic UMI filtering. Here you can see the distribution of the number of reads per UMI. This is a common way when you see 2 peaks on the distribution. And usually the left one is the one that contains erroneous UMIs that contribute to artificial diversity, and the true molecules are on the right. And MiXCR automatically sets the threshold. It means that all the UMIs that are covered by the less number of reads than the threshold will be excluded from the analysis. And we have the same plot for every sample. Now, this is a very useful plot which can, for example, tell you if the sequencing depth was enough for the diversity that you have in the sample. Now, the last metric that I want to show you today is the chain usage. Again, I'm going to use the command, and the metric here will be chain usage. Now, here, VZJCA files or CLNS files are not the same. If you're going to use the VZJCA files, then you're going to see the percentage for every chain in the numbers of alignments. And if you're going to use CLNS files, then you'll see the percentage of assembled clones that contribute to a specific chain. I'm going to go with CLNS files again, and the output file name is chain usage.pdf. Now open. So here you can see the distribution of the clonotypes in the sample. We right away see that about 10% in every sample is occupied with the heavy chain, the rest of the sample is the light chain, and they're pretty much similar proportions for two types of the light chain. You can also see what portion is occupied by the non-productive clonotypes, like the ones that are out of frame or have stop codons in it. We see that only immunoglobulin chains are present here, which means that there were no contamination by T cells, for example. And I'm also going to put a link to the documentation portal where you can see some parameters that you can use to adjust the plots that I've just shown you. That's it for today, and thank you for watching.